Hi everybody, Dr. Ahles here. In this short video, we're going to talk about the next macromolecule in the human body, and that is proteins. Proteins make up about 10 to 30% of your cell's mass. If we're considering how much space that is or how much mass that is, this is the most abundant macromolecule after water. So we've got a ton of water in the cell, then we've got a ton of proteins. When we talk about proteins, just like our other organic compounds, remember they're absolutely going to have carbon, that makes them organic, but also that hydrogen and oxygen, like we saw with carbohydrates and lipids. In proteins, we absolutely see the addition of nitrogen, that's going to help us to build our protein bonds, and some of our proteins will also include the element sulfur. So adding in these two new elements completely changes the structure and the function of the organic molecule, making it a protein. You will see the terms protein and polypeptide used interchangeably. So what I mean by that is polypeptide means the same thing as a protein. All of my proteins, or all of my polypeptides, are made out of long strings of amino acids. So amino acids are the monomers, the little pieces, that make up my big pieces, my polymers, called proteins. When we look at an amino acid, it got its name because it has an amine group on one side and a carboxylic acid group on the other side. The acid group and the amine group are what we use to connect these amino acids to each other. And this little group that we see up here, represented with the letter R, this is the functional group of the amino acid. When we think about the functional group of an amino acid, I like to call it the personality type of that amino acid. Amino acids aren't introverts or extroverts or, thinking or feeling. When we talk about the personality type of an amino acid, we're talking about, are you polar? Are you non-polar? Do you have a negative charge or a positive charge? That's what this R group tells us. Basically, all of your functional properties. What do you do with water? What do you do with your neighbors? When we're talking about building proteins, connecting together the different amino acids, we use something called peptide bonds. Let's underline, highlight, star peptide bonds. Like I mentioned way back with our types of bonds, peptide bonds are only found in proteins. This is how we connect amino acids to one another. Amino acids come together with these peptide bonds to make us these big proteins. So we build those bonds like we talked about before with dehydration synthesis. We're pulling water out, leading to the formation of a peptide bond. And we break them using that process called hydrolysis, where we add water and separate amino acids from one another. When we talk about different sizes of protein polymers having more than one amino acid, we've got several different terms that we'll use for them. A dipeptide is made out of 2 amino acids, like di says. Tripeptide, made out of 3 amino acids. A polypeptide is a string of amino acids, up to about 50 of them. Once we have more than 50 amino acids grouped together, we're going to call that a protein, not just a polypeptide. So when I use the word protein, consider that means I have over 50 different amino acids that are all put together to form that structure. So we go from a single amino acid into some kind of peptide, maybe a dipeptide, all the way up here to a functional protein made out of many, many different amino acids. When we think about proteins, let's underline, highlight, star that the order that the amino acids are present in is going to be really important. If I change the order, if I put the wrong amino acid in, this is going to give me big problems when we're thinking about the activities or the functions that the protein is supposed to do. So when I look at proteins, we have 4 different levels of chemical structure. The first level, the most basic level, is what we call the primary structure. And what I want you to underline, highlight, star about primary structure is that this is the sequence that the amino acids are connected in. So the sequence or the order of those amino acids, that's my first most basic level of protein structure. We start there because if we change this primary structure, the first level, all other levels of protein structure will not develop correctly. So I've got to start correct at the very beginning. I've got to start with the right sequence of amino acids. Once I got that correct sequence of amino acids, my amine groups with the nitrogen and my carboxylic acid groups bond to this oxygen will interact with each other to make things called an alpha helix or a beta sheet. I like to think about an alpha helix and a beta sheet. The alpha helix is kind of like those curly ribbons you put on a birthday present. So the amino acid chain just circles around and around and around itself all the way down. That's an alpha helix. Versus a beta sheet is like if I took a string of beads and I curved it back and forth in a zigzag pattern. That's a beta sheet. Alpha helices and beta sheets form because I have polar amine groups and polar carboxylic acid groups. I'm interacting these structures just like I did to connect them to each other. Because I take those alpha helices, here's that curly Q, I take those beta sheets with these flat arrows. When I squeeze them all together to make something that's 3D, now I've made what's called tertiary structure. So tertiary structure is my overall 3D shape. All parts of that amino acid chain have folded together. Notice that this is the most common level of structure that proteins in our body will get to. So we have an order, a sequence of amino acids, primary structure. We do some curly Q ribbons or some zigzags back and forth. That's secondary structure. When I fold all of those things on each other, now I've got this tertiary structure. The last kind of structure that I can have in proteins is what we call quaternary structure. And quaternary structure requires 2 or more polypeptide chains. I want you to underline, highlight, star 2 or more. You cannot have quaternary structure, this biggest level of structure in proteins, unless you have more than 2 polypeptide chains. So 2 or more chains leads to this quaternary structure. I took all that time to talk about the levels of protein structure because when we think about proteins, their anatomy, their shape, needs to be correct for them to be able to perform their physiology, their function. So there are 2 main anatomies or 2 main protein structures that we see in the body. The first are what we call fibrous proteins. So when you think of a fibrous protein, just like its name suggests, these are long and stringy. They're protein fibers. We also, though, have proteins that are called globular proteins. So over here, we're seeing one that's called an immunoglobulin, or an antibody for the immune system. Here on the right, we're seeing the hemoglobin protein in your red blood cells. Notice that it's made out of 1, 2, 3, and 4 different polypeptide chains. So we have quaternary structure on these because they have more than 1 chain. And then we have these long, stringy, fibrous proteins that we see. It's possible for a protein in your body to permanently lose its shape. That would mean that protein is called denatured. So the best example of denaturing that you're familiar with is when you cook an egg in the pan, the whites of that egg start out clear, but once you heat them up, they completely change their structure, which means that now light can't pass straight through them. So denatured proteins permanently lost their shape. And remember from our statement up here, if I lose my shape, if I lose my structure, I also lose my function. I lose my ability to work. So let's do a little compare and contrast between my fibrous proteins and my globular proteins. When we talk about fibrous proteins, these are those ones that we said were long and stringy. Compared to my globular proteins, many of them are spherical or they look like little clumps together. Fibrous proteins are my structural proteins. Either they're giving shape to my cell as a whole, or they're really important for a specific action inside that cell. Compare that with globular proteins. These are my functional proteins. So look down here at our examples. Things like antibodies that run the immune system. Things like enzymes that get everything done. Things like receptors that listen to the outside world. Globular proteins do a lot of different things compared to my fibrous proteins that are really all about structure, all about shape. Fibrous proteins are insoluble in water, meaning that they do not dissolve, they won't float around, whereas globular proteins tend to be more soluble. They play nice with water a little bit better. A benefit of my fibrous proteins, my structural proteins, is that they're very stable. Globular proteins are a little more prone to being denatured, just because their shape isn't nearly as stable or as structural. Now, when I look at these types of proteins, notice that several of these proteins are things we're going to talk about later in our semester. So collagen, very important in many of the types of tissues we're going to talk about. This is a really strong protein. Same thing with keratin. This is the protein that makes your skin tough. Actin and myosin help us with the process of muscle contraction because these long strand-like fibers pull right past each other. So big picture, fibrous proteins, they're going to give us mechanical support, meaning they're going to help us to shape our cell. They have tensile strength, which means they're not going to fall apart, and contractility. That's the ability to do muscle contractions. Whereas globular proteins can do pretty much anything. They're involved in signaling or your immune system. Really, if you want to think about what globular proteins do, they help us maintain homeostasis. They keep us at balance in the body. So when we're looking at our proteins, notice that we have functions that fibrous proteins are better at here on the left, and functions that globular proteins are better at here on the right. For all my visual learners, now you can see those different kinds of functions and kind of put them together with a picture. The last thing I want to land on, though, for our discussion of proteins is this right here. Proteins acting as an enzyme. When we talk about enzymes, these are globular proteins that we're going to use to help a chemical reaction occur. Remember that when we were talking about chemical reactions way back at the beginning of these videos, chemical reactions have this thing called activation energy. It's that hump you have to get over to start something. When I don't have an enzyme to help to give that activation energy, notice how big that hump is. If I add that enzyme, the enzyme makes it easier for the chemical reaction to occur, so I don't need as much energy to get to that point. So because enzymes decrease the amount of energy that's required to start a chemical reaction, we would say that they increase the speed that the chemical reaction happens at. If it's not going to take me as much energy to start this reaction, then I'm able to do it more quickly and more efficiently. Enzymes are reusable. So not only do they drop that energy, I can have them do it again and again and again. Functioning as enzymes, perhaps the most important thing that enzymes do, or that proteins do in your body. So, a couple of characteristics of those enzymes. Number one, they're very specific. Enzymes have what's called an active site. This is where the things they're working on can actually attach to. If you don't fit in my active site, if instead of being an amino acid, you're a monosaccharide, I'm not going to do anything with you. So they work with particular substrates. They're very specific. We can often tell what those substrates are based on the name of them. So for example, an enzyme that's going to work on a protein or a dipeptide might be called a peptidase, or an enzyme that might work on a sugar. We could have that called galactose that breaks down galactase, that breaks down galactose sugar. So if you see a name that looks really similar to something else you know, that's not a coincidence, that is on purpose. So enzymes, very specific, do a particular job, and because they're so specific, we can name them based on that.